Hey everyone, welcome to this week's module. We're going to be starting off our 2-week look at the nervous system here. This week we're going to be looking specifically at what makes up our nervous tissue, and then how you take those tissues together and you make up one of the two organs that makes up the central nervous system, which is the brain. Okay, then next week we're going to finish off our discussion looking at the spinal cord and then everything else that extends off of both the brain and the spinal cord. So with the nervous system, you can divide it into two main divisions. So we have the central nervous system, which is made up of brain and spinal cord only. Okay, don't ever get that confused. I can throw in anything else to distract you, but remember, brain and the spinal cord are the only two organs that make up that central nervous system. Okay, everything else that extends from both the brain and the spinal cord are part of the peripheral nervous system. All right, and this includes both the voluntary effectors that are controlled by the somatic division, and then everything else that's automatic. Okay, and we've looked at both voluntary and involuntary control when we talked about the different types of muscle tissue. Okay, so let's think back to the muscular system. When we looked at the differences between skeletal, smooth, and cardiac muscle, remember skeletal muscle is voluntary, right? We control that. So we make a decision in our brain that we want to say, I don't know, lift a table. And so that is going to then send a motor signal through a somatic motor nerve to whichever muscle needs to start to work. In that case, say you're lifting something, it's going to be your bicep brachii. So the brain's making a decision, sending a signal out, controlling that movement, getting that bicep to work voluntarily. Okay, that is controlled by the somatic division. So when we refer to somatic nerves, we're referring to those that connect to structures that we are voluntarily controlling. Okay, so in contrast, the autonomic nervous system, which the two divisions are the sympathetic and parasympathetic, the autonomic, think automatic. Okay, so anything else that is controlled involuntarily, such as your glands, your smooth muscle, your cardiac muscle, all of those structures are going to be regulated automatically by the autonomic nervous system. So together, somatic and autonomic make up your motor and sensory nerves of your peripheral nervous system. Now, with this autonomic division, we're going to focus on this towards the end of next week's lecture. But for now, just know that the two divisions are the sympathetic and the parasympathetic. Okay, so sympathetic, it's your nervous system's response to stress. Okay, so that's why it's known as your flight or fight response. So an example here, if I was being attacked by a bear right now as I'm recording a lecture, I would be extremely stressed out and I would be trying to run away from this bear that got somehow into my house. Okay, that would be an example of your sympathetic nervous system kicking in. I hope some of you have been able to activate your parasympathetic nervous system, right? By somehow relaxing, forgetting about what's actually going on out there in the world and just being able to just rest and take some time for yourself. The way I want you to think of these two, sympathetic, parasympathetic, is that they're both always going to be somewhat active, but based upon circumstances, based upon physiological activities, they're going to become more active, one more than the other at different times. So if you're stressed out and your body needs to do what it needs to do to respond to that stressful environment or response, then your sympathetic is going to kick into high gear. Okay, so let's think about that. What's going to happen when you're stressed out? Your heart rate's going to kick in, your breathing rate's going to increase, your pupils are going to dilate because you're going to be trying to take in more visual information, right? So when you get stressed out, the sympathetic division is the autonomic division that kicks in to be able to increase activities, specifically of the heart, your breathing. There's a whole list we'll go into next week, okay? But think about sympathetic is your flight or fight, your stress response. Now, in contrast, parasympathetic, what happens when you're relaxing, right? When you're relaxed, your heart rate goes down, your breathing rate goes down, okay? It's the opposite. So the parasympathetic, if you're resting or digesting, then you're going to see the parasympathetic nervous system is going to be increasing activity. So your heart rate would go down, breathing rate goes down, digestive organs would start to function. If you're stressed out, that's going to inhibit your ability to regulate the function of those digestive organs as well. All right, so sympathetic, think stress, flight or fight response. Parasympathetic, what's going to happen when you're resting or digesting. Okay, and we'll get into that in a lot more detail next week. So for now, Just make sure you have a clear understanding. You have the two main divisions of the nervous system. So central nervous system, which is your brain and spinal cord only. Everything else gets classified under the peripheral division. Okay. And within that peripheral division, we have the somatic division, which is voluntary control, and then the autonomic division. Okay. And so somatic, think somatic motor, right? And then autonomic motor. Your sensory division falls under this somatic division, right? So any sensory information that you receive into your body falls under this somatic division, okay? And then finally, automatic control, you have the sympathetic division, which is flight or fight, your stress response, and then parasympathetic division, which is rest or digest. Remember, the peripheral nervous system is every nerve that is going to somehow connect the brain or spinal cord with your muscles, glands, or any of your other sense organs. Okay, so with sense organs, we're looking at specific sensory receptors. We're going to be detecting, say, light touch, right? We talked about Merkel cells, for example. That was an example of a sensory receptor in our epidermis. Okay, so Merkel cells detect light touch. That information is going to be sent in. So you're detecting light touch here. It's going to be sent in through sensory nerves. And where are they going to go? They're going to go into the spinal cord to be sent up to the brain to be dealt with, right? That's where we interpret this information. So you have sensory going through the somatic sensory nerves into the spinal cord, up into the brain, and then finally your brain then is going to determine, okay, well, what do I do with this sensation? If it's, say, a spider you're feeling, you're going to have some fear and probably want to swat it off of your hand, right? So that's going to then kick in some of the motor somatic fibers then to activate maybe your other limb to go and swat that spider that was, causing that sensation on your finger. There's so many complex pathways and mechanisms involved here. This is just one example that I'm giving you. I would highly recommend as well, before going any further, take a look at the crash course video, which gives you another sort of animated perspective here on your divisions of the nervous system. It's the first one. It goes into a brief overview and then also kind of goes into nervous tissue as well. Okay, so central nervous system, peripheral nervous system. We're going to spend pretty much the rest of today focusing on the brain, and then next week we'll get into everything else. You have incoming and outgoing information, right? So with the brain, you can see here that we have the extensions from the brain being the cranial nerves, okay? And from the spinal cord, the spinal nerves. So we're going to be receiving sensory information is going to be coming in to those cranial nerves. There's certain ones that are sensory. And then that information is then going to go into the brain directly through those nerves. Okay, one example would be your second cranial nerve, your optic nerve, right? We receive visual information in through our eye, which gets transmitted and passed along to that cranial nerve number 2. That cranial nerve then sends that sensory information directly to the brain. Okay, so when you're looking from the periphery centrally towards brain, that's where you're looking at sensory. Okay, specifically here with the pathway to the cranial nerves being sent directly to the brain. Now, we can have peripheral sensory information everywhere else in our body, and those sensory nerves are then going to send signals through the spinal nerves. So for the example that we talked about, if you're feeling any kind of sensation here in your periphery, that's going to be sent through one of the spinal nerves, that contains sensory information, which is then going to synapse here in the spinal cord. So that's where you're seeing from spinal nerves to spinal cord. And finally, that information then needs to be sent to the brain, right? So that's what this flow chart is showing you. Now, once you've received that sensory information, then we are going to be controlling some sort of somatic or voluntary motor as a result of that. In some cases, though, like thermoreceptors, for example, baroreceptors we've talked about, right? You receive incoming pressure information from your aorta or carotid bodies, and then that information is sent to your brain. Your brain then is going to send automatic signals back to your heart to be able to regulate your heart rhythm to help control blood pressure, right? That would be an example of autonomic regulation there. Vice versa, if with the spider example, you feel something, and then as a result of seeing it, and also feeling it, you're going to send motor signals likely to maybe this limb to shake it off or to this limb to go and hit it off, right? That's an example of then somatic voluntary information that's going to be sent out, okay? So this is just showing you here, sensory information is coming in towards the central nervous system, whereas motor information is leaving the central nervous system and exiting into the peripheral to then have its effect. Now, within our nervous tissue, just think back, right, to the levels of hierarchy, right? Our most basic level was chemical. And then one step up, we are all made up of cells, right? So we're going to be looking at those cells. And remember, we put together specialized cells to make the tissue. So within our nervous tissue, we have two different types of cells. We have neurons, which are our nerve cells, and then we have glial cells, which are also known as neuroglia. Now, the neurons, these are the ones that are actually sending and transmitting that information, right? When we talk about the whole role of the nervous system is to communicate and send signals to be able to activate effectors, the signals themselves that are being sent are sent via the neurons. Now, in contrast, the glial cells, these are there to help support the neurons, okay? So the glial cells in and of themselves, here's a few examples, your oligodendrocytes, microglia, astrocytes. There's others as well, but the whole role of the glial cells is to be able to support the neurons. Now, with the neuron, remember your generic cell, right? You have a plasma membrane, you have then your nucleus and your membrane-bound organelles inside. That's your generic animal cell, right? And then we've looked at different specialized cells. We looked at specialized muscle cells, how they were different. We looked at specialized epithelial cells. Now we're going to be taking a look at the neurons, the ones that make up our nervous tissue. So similarly, you can see here, located within the cell body of a neuron, you have all of those membrane-bound organelles. So everything that's going to help this whole cell, the neuron, to be able to function is located up here within the cell body. Now, extensions from that cell body, which are there to receive incoming information, those are known as the dendrites. I want you to picture this almost as me standing up in front of you, okay? So here's my head. Dendrites are my hair, which has not been cut. It's quite interesting at this point. The dendrites are receiving incoming information, incoming signals, here to the cell body, which then passes that information along to the axon, okay? So the information is always going to flow in through the dendrites, through the cell body, through the axon to the terminal end, okay? And at this point here, terminal end of the axon, these are known as the terminal branches, right? And this is where you're going to see the neuron connect either with another neuron or connect with a effector. So what's an example of an effector? A muscle cell, right? We looked at the neuromuscular junction where we saw the terminal end of a neuron, specifically a motor neuron, in that case, would connect with the muscle cell and send that signal to be able to activate it. Go back to that muscular lecture. Remember we looked through transferring the signal, right? So you'd see how the nerve signal was sent from the brain through the motor neuron, ultimately synapsing there at the neuromuscular junction to cause the muscle cell to contract. And that's the whole point, right? Think of this neuron as like an extension cord. So you have the male end of the extension cord that you're plugging into the source, and then ultimately that extension cord is going to transmit the electrical signal to its destination. So we'll look at the various ways in which neurons can connect to different parts of the body and also to other neurons. Your main three parts: cell body, the dendrites, which are like the hair, and then the axon, which would be sort of just the rest of the trunk and torso down to the terminal branches. Now, depending on the length of the neuron and where it's located, lots of neurons are myelinated, which means that they are wrapped in this fatty substance known as myelin. And myelin is there to act as like an insulating sheath. When we're sending these electrical signals, for example, a motor neuron, we're looking at one neuron that's going all the way from your spinal cord, all the way, say, down the length of your leg or down your arm. Some of them are very long and have to transmit these signals over very long distances. And so to do so and to make that process more efficient, our bodies are set up where these neurons are myelinated, where they're wrapped in this layer of myelin to be able to provide that insulation to help to, number one, make sure the signal is consistent down the length, but also make sure the speed of transmission is as efficient as possible. All right. And this myelin is made by specific cells, which we find known as Schwann cells. Okay. And here's an example here of a nucleus of one of those Schwann cells. And that Schwann cell is going to be producing that myelin, which then wraps itself around the neuron. Between each of these Schwann cells, you have these gaps, right? If we look back to this picture here, you can see it pretty evident, right? I always refer to Schwann cells as like the hot dog buns, all right? So you have hot dog buns that are wrapped down the length of the axon. So between each of the hot dog buns, between those Schwann cells, those are regions that are known as nodes of Ranvier. Now, these spaces are essential. You can see them here, and all these linear portions. Okay, those are all the nodes of Ranvier. Now, at these portions, this is where we have voltage-gated channels that allow us to create a new signal to be sent to the next node, which is then recreated and sent to the next node. Okay, as we get into talking about action potentials and how they are produced and how the signal is essentially transmitted throughout the neuron, it'll become more clear. But for now, as long as you understand the anatomy, we can have myelinated to increase the speed of transmission. Okay, so myelinated with the Schwann cells and the nodes of Ranvier, or you can have unmyelinated axons. Now, as I mentioned at the beginning, we've taken a look at sensory information coming in and then motor information going out from the central nervous system. So we can classify those neurons based upon new terminology here, and that's based upon the direction where that impulse is going to be traveling. So for sensory neurons, the signal is going to be coming from the periphery in towards the central nervous system. Okay, so those sensory nerves are also known as afferent nerves. So the key here, afferent, think arriving, right? We talked about this when we went through the lymphatic system, right? Afferent vessels were arriving at the lymph nodes, right? So here it's the same kind of situation. So the sensory neurons, the impulse is going to be coming from the periphery. So say like a touch receptor in your hand, signal is sent in towards the central nervous system and it's arriving at the central nervous system, okay? So sensory are also known as afferent. And then in contrast, motor neurons are also known as efferent neurons, right? The signal here is coming from the central nervous system and exiting out to the periphery of the body. So afferent or efferent, motor neurons are efferent and they are exiting the central nervous system. Now, within the spinal cord, we're going to see how interneurons are responsible for connecting the sensory to the motor in some cases directly, also to send impulses. Let's say sensory information is coming into the spinal cord. Once the information is in the spinal cord, well, then it needs to be sent via interneurons to get it through the central nervous system to wherever it's going to be processed. And then finally say that sensory information is going to go to the brain. Then there's going to be other interneurons connecting that back to a specific motor neuron. Okay, so those interneurons are connecting neurons and they conduct impulses from sensory neurons to motor neurons. And we're going to see the difference between your monosynaptic reflexes and polysynaptic reflexes. Okay, and it just means that there's more connecting neurons in polysynaptic reflexes than there are in monosynaptic. As I mentioned before, we have these neuroglia cells. They are the most prominent type of cell within our nervous tissue, and they're there as the neuron helpers, right? They're there to provide support, and they each have a different role. Oligodendrocytes, for example, they are found only in the central nervous system, and their main job is to wrap myelin around the axons of neurons in the central nervous system. Okay, so think about it. If we're in the central nervous system, what two organs could you be looking at? Brain, spinal cord. That's it. So within the brain and the spinal cord, oligodendrocytes do the job of producing myelin. In the peripheral nervous system, it's those Schwann cells that are doing the job of producing that myelin. With the neuron helpers, they are much more numerous than the neurons. In the brain, I think we have about 100 billion neurons compared to about 10 trillion glial cells. So the oligodendrocytes are responsible for wrapping myelin around the axons within the central nervous system. We also have astrocytes, and they are key for connecting blood vessels within the nervous tissue of the central nervous system to the neurons themselves. They act as the blood-brain barrier. One molecule that is able to readily pass from the blood to affect the nervous tissue is alcohol, and that's why it has such a profound and immediate impact on our nervous system. Astrocytes, though, are there to be able to regulate and inhibit what passes from the blood into our nervous tissue. And then lastly, but definitely not least, microglia, which think microorganisms. Okay, they're there as specific neuroglia that provide immune support within our nervous tissue. And then lastly, we talked about within the peripheral nervous system, we have the Schwann cells. So the oligodendrocytes in the central nervous system and the Schwann cells within the peripheral nervous system are doing the same job, just in different compartments. Okay, so they're both going to be producing the myelin that is going to be myelinating specific axons. Within the peripheral nervous system, we are going to be looking next week at all of the different peripheral nerves. We're going to be looking at spinal nerves and cranial nerves. The spinal nerves originate from the spinal cord. The cranial nerves originate from the brain. And these are the peripheral nerves within that peripheral nervous system division that will travel throughout the body to whichever destination they are to innervate. Now, with these Schwann cells, remember Schwann cells are located in the peripheral nervous system, and they are there to produce the myelin that insulates the neuron, and the gaps between those Schwann cells are known as the nodes of Ranvier. And the whole point of myelination, whether it's the central nervous system or the peripheral, to increase the speed of transmitting that specific nerve signal. Now, you may think that this looks similar to something we looked at previously when we looked at how a muscle was made up. Bundles within bundles within bundles. Well, we have the same type of setup where all of our neurons, they are all wrapped within a connective tissue sheath that's known as the endoneurium. Okay, so here we're looking at a myelinated axon. Okay, that's just one neuron. That myelinated axon is wrapped in endoneurium. You take a whole bunch of them, you wrap them each up individually in endoneurium, you bundle all those up together, and you create a fascicle. Okay, and that fascicle is surrounded by perineurium. Okay, and then finally you take the fascicles, bundle them up together along with your arteries and veins, and those then are what form the nerve itself. Okay, so these bundles of peripheral axons that we find in the peripheral nervous system, highlight that, okay? This is how a peripheral nerve, whether it's spinal or cranial nerve, this is how it is set up. Hopefully this will help in understanding how we can have both sensory and motor components within a single spinal nerve. Okay, and we'll talk about that next week when we look at both cranial and spinal nerves. So you have the axon itself, so one single neuron is going to be wrapped in endoneurium, then multiple neurons bundled together become the fascicle, and that fascicle is wrapped in the perineurium. And then a number of fascicles then all bundled up together by epineurium is what produces your peripheral nerve. Now, if you haven't stopped to watch the video yet, I would highly recommend that you just put this on pause and go and take a look at the video. I would look at the crash course video that goes over action potentials, as well as there's an individual video looking at action potentials. So action potentials, okay, essentially that just means nerve impulse. All right, this is the means by which a neuron is able to transmit an electrical signal. So the action potential is that nerve impulse. It's that nerve signal that's being sent from the cell body through the axon to the terminal end branches. Now, something we previously discussed is how the extracellular and intracellular environment have different concentrations of ions. Okay, and two that we focused on were the sodium and potassium ions, and those concentration differences are regulated by the sodium-potassium pump. So if you recall, the sodium-potassium pump uses active transport mechanisms to be able to pump sodium out of a cell and to bring potassium into the cell. The reason why is so that we can open up specific channels within that cell membrane and then allow for sodium to rush in and potassium to rush out. And we're going to see how this applies specifically to action potentials as we move forward here. So first off, you need to remember sodium is very high in concentration in the extracellular space. So what's outside of the cell? That's your interstitial fluid. Right? When we look at tissues, remember tissues are made up of cells. Between those cells, you have your interstitial fluid. Inside those cells, you have your cytoplasm. Okay, so in the interstitial fluid, you see an abundance of sodium ions as well as an abundance of calcium ions, chloride as well. Within the cytoplasm, this is where you have an abundance of potassium ions and other negative charged proteins. Now, when you think about this, if you have a lot of sodium outside of the cell and you put a specific sodium channel right into that membrane, which means you're opening it up only for sodium, which direction do you think the sodium is going to move? From outside to in or from inside to out? It's going to move from out to in. So sodium is always going to rush into the cell. And then similarly, if you put a potassium channel, which is specific just to allow potassium movement, potassium is going to want to rush from its area of high concentration inside the cell to the external environment, right? So if you have sodium channels, the sodium is going to rush into the cell. Potassium channels, potassium is going to rush out of the cell. Utilizing these and activating them at specific voltages is how we are able to transmit these electrical signals, the action potentials, throughout the length of the neuron. At rest, you have an abundance of sodium outside the cell and you have an abundance of potassium inside the cell. Now, because you have also a number of negatively charged proteins inside the cell, that means that you are going to have an overall resting negative membrane potential. when the cell is at rest and the sodium potassium pump has done its job at making sure that there's an abundance of sodium outside the cell and an abundance of potassium inside the cell, the net is going to be a negative overall charge, okay, to the cell. You do not need to know these numbers, and you do not need to remember this chart. The reason I've added this to our lecture is to be able to give you a little bit of hopefully understanding into what's actually going on as an action potential occurs. At rest, the membrane is going to be holding a charge of about negative 70 millivolts. There is going to be allowed sodium to enter into the cell. So think about this. Sodium, which is high outside the cell, is allowed to leak into the cell. That is going to be causing the inside of the cell to become more positive. Now, an action potential is an all or none phenomenon. So it either happens or it doesn't. So if there is enough stimulus causing enough sodium to leak into that cell, it's going to reach this threshold level. And as soon as it hits negative 55, negative 50, negative 55, that is the trigger for opening those voltage-gated sodium channels. And as soon as those voltage-gated sodium channels open, sodium rushes into the cell. And so sodium's positive. If it's coming into the cell, that's going to make that inside of the cell very positive. Now, at this point here, you've had so much sodium rush in. At positive 30 millivolts, the sodium channels get closed, and now the potassium channels open. And so let's go back here. If now the potassium channels are opening, those potassium are going to then flow out of the cell. So think about that. If you have a whole bunch of positive ions leaving the cell, that is going to cause a huge drop, right? So you're now going to be repolarizing when those potassium ions leave the neuron. So let's just look at these two events. Sodium is rushing into the cell first. That causes that depolarization. And then at positive 30 millivolts, potassium gates open and potassium now rushes out of that neuron. Now, at this point here, you now have excess amount of potassium has left the cell. And so the sodium potassium pumps then do their job to try to regulate those levels again and bring that back to a resting membrane potential of negative 70. Okay? This whole event of depolarization, repolarization, hyperpolarization, that is an action potential. Okay, so let's go back. When we're looking at that, at each node of Ranvier, this is where these channels are located. You're going to have an action potential start here at the beginning of the axon. That's going to cause an action potential to be created. That action potential is then going to zip along to the next point where there are more channels. It will then depolarize those channels, which then kicks off this whole process again. Okay, so each time you're going to have another action potential created. It's going to be the opening of sodium channels followed by the opening of potassium channels in conjunction with one another that's going to cause these electrical events to occur. All right. And so then as you look at one of those myelinated here, for example, myelinated neurons, at each node of Ranvier, this is where you're going to have new action potentials created. So the action potential first created here is going to then zip along because it's insulated, right? So that electrical signal is going to transmit through the myelinated region. And then here at the node of Ranvier, it's going to then stimulate and activate brand new sodium channels to kick off a new action potential. And then from this node of Ranvier, that new action potential is going to zip along to the next node. New action potential is created, zips along to the next node. So in a myelinated neuron, this is what helps to speed up transmission because pretty much the action potentials jump from node to node to node, okay, helping to increase the speed that this action potential propagates down the length of the neuron. Now, your textbook gives you this example here, okay? We're still looking at the axon, right? So we're looking at the nerve cells. Here's our cell. There's your cell membrane. Okay, remember the resting membrane potential is going to be overall negative. So some sort of stimulating event is going to be causing a depolarization, which causes the leak of sodium into the cell. The inside of the cell is becoming positive. That then creates the action potential. Okay, this is where then this information can be overlain on this diagram. Okay, so now here you have the threshold potential has been met. You have depolarization followed by repolarization. And then this stimulates the next adjacent region, which you can see happening here. Depolarization followed by repolarization and so on. Every time you have that depolarization event, it causes an increase in positivity to that region. So if you have an unmyelinated neuron where you have just channels next to channels next to channels next to channels, the depolarization in this region, which makes it more positive, is going to cause this region to become more positive. So as that happens, then this reaches threshold potential, and now a new action potential is created, which then causes the next adjacent region to reach threshold potential, and another new action potential is created, which you can see here why you're literally having to depolarize, 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 all the way down that whole length of the neuron. So when you myelinate a neuron, now you just have to have action potential created, so depolarize, repolarize, depolarize, 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 depolarize. Okay, so it's much more efficient than going down the length of the neuron. One thing to remember about action potentials, they can only move in one direction. Okay, so that's very important. So originating here near the cell body and then terminating down near the terminal end branches. The key summaries here are in the following slides. So at rest, the inside of the neuron is negative, right? You know that now, due to an excess of sodium outside of the cell. Now, as soon as you have that triggering event, that's the depolarization. So the voltage-gated sodium channels open and sodium is going to rush in. Okay, there's an acronym I like to use, SYN and POUT. Okay, so SYN, S-I-N, sodium always moves in, and POUT, P-O-U-T, potassium always flows out. Okay, so always think of that whenever you're trying to figure which direction those ions are going to be moving. So the stimulus is going to trigger the opening of sodium channels. When sodium channels open, sodium is going to rush into the cell. Now, as that happens, the sodium then depolarizes the membrane. The inside becomes more positive. Once it hits that positive 30, then you have the potassium channels opening, and the potassium channels opening causes potassium to rush out, right? SYN and POUT. So potassium is going to rush out. That's the repolarization. That depolarization followed by repolarization, that is your action potential. Okay, and it's that depolarizing event that happens that then causes depolarization of the next immediate region. All right, and that's how the signal is transmitted down the length of the neuron. Okay, so one little added point here, we're looking obviously at a myelinated neuron, right? So here you can see there are your Schwann cells, you have your myelinated regions, and between those you have your nodes of Ranvier. Okay, and remember the nodes of Ranvier, this is where your voltage-gated channels are. So your sodium voltage-gated and your potassium voltage-gated channels are located here. Okay, so initial stimulus causes an action potential. That action potential jumps to that next node of Ranvier. Here, it's going to depolarize the sodium channels, which are then going to cause repolarization by the potassium channels opening. So you have another action potential created here. That signal jumps then to the next node. Okay, new action potential created. Jumps to the next node. Okay, so this jumping from node to node is a phenomenon known as saltatory conduction. All right, and this only happens within myelinated axons. All right, in unmyelinated axons, remember, each depolarizing event is going to impact the next adjacent region. Okay, so much slower, much more inefficient in an unmyelinated axon. In a myelinated, saltatory conduction allows for that action potential to jump to the next node of Ranvier. Now, anytime you have a neuron connecting with another neuron, that region is known as a synapse. And when we get into looking at the autonomic nervous system next module, you're going to see that there are always in the sympathetic and parasympathetic divisions, there are two neurons involved in transmitting the autonomic signals. So the first one, which is before the synapse, is known as the presynaptic neuron. And the one located after the synapse is known as the postsynaptic neuron. Okay, so for now, we're just focusing on terminology. Next week, we'll get into the specifics and the nitty-gritty. Within this connection, the synapse, we're going to have the presynaptic neuron, which exists before the synapse, and then you'll have the postsynaptic neuron, which exists after the synapse. So the synapse is the connection that the pre makes with the post. All right, and the structures that are connecting, you have the presynaptic end bulb, which is the end of that terminal branch from the presynaptic neuron. Connecting, you have this gap between, known as the synaptic cleft, and then you have the postsynaptic end plate or plasma membrane. Okay, and there you can see that you have specific ligand-gated channels, which are responsible for binding the neurotransmitter that's released from the presynaptic, which then cause them to open and cause a whole new signal to be created in the postsynaptic. before, like I use the analogy of think about like an electrical cord. Well, here it's not that we're literally plugging one electrical cord into the other. Here you have an electrical signal that ends up being converted to a chemical signal, the release of the neurotransmitters, which then that chemical signal causes for a new electrical signal to be created in the postsynaptic neuron. And then that signal sent throughout that postsynaptic. Now, there's also a video on these reflexes, so I highly recommend going to that as well to give you a bit of a visual. Reflexes are set up in two ways. We have monosynaptic reflexes and polysynaptic reflexes. So with monosynaptic reflexes, these are the ones that are really, really quick, and these are almost primal or more primitive reflexes. With polysynaptic just take a little bit longer. With polysynaptic, there are at least 3 neurons involved, so more synapses, hence the name polysynaptic. The two examples we're going to take a look at, monosynaptic reflex that we're going to look at is the knee-jerk reflex. If you go to get your reflexes tested, they take the reflex hammer, tap it on your patellar ligament, and that causes your quadricep to contract and kick out your leg. We're going to look at that. Okay, so that's a really quick reflex. A lot of your tendons, so your patellar tendon, hamstrings tendon, as well as your Achilles tendon, you can test nerve conduction by simply tapping on them to see those monosynaptic reflexes. Now with polysynaptic, the example we're going to talk about is the withdrawal reflex. And so that's, say you put your hand on a hot stove, all right? Before long, you are going to just be whipping that hand off of there, right? You're going to have that sense of pain that you're going to be perceiving, that ability for yourself to be able to quickly withdraw your hand. That's a polysynaptic reflex. All right, so let's first take a look at the monosynaptic reflex here. So within our muscles, we have specific receptors, right? We talked about thermoreceptors, we have mechanoreceptors, proprioceptors. These stretch receptors would be an example of mechanoreceptor. By tapping on the patellar ligament, that causes a passive stretch within the quadricep. So that sends a signal. Remember, think about what type of signal is this if it's incoming, right? It's from the periphery, incoming into the spinal cord. So that is a sensory signal, okay? So the stretch receptor converts that information and sends it into our central nervous system via an afferent sensory neuron. That sensory neuron then arrives at the spinal cord, okay? So it's passed through the spinal nerve. And now as it's arriving at the spinal cord, it passes through a region known as the dorsal root, okay? So sensory information always comes in through the dorsal, so the back of the spinal cord. That sensory neuron, remember it's monosynaptic, is going to directly synapse with the motor neuron that's going to be sending a signal out. And what ends up happening, think about this, when you have your patellar reflex tested, you tap on the patellar ligament and that causes your leg to kick. So that small stretch sends a signal through the dorsal root, which then synapses directly. So there's your monosynapse, which synapses directly to the motor. And the motor always leaves the ventral portion of the spinal cord, which we'll see next week. So then that motor root leaves the ventral portion, which then causes your quad to contract. All right, so that's one example of a monosynaptic reflex. So any of the other deep tendon reflexes would work in the same way. So a couple of key points here. The sensory information, afferent, right? The afferent information is coming in, arriving at the central nervous system through the dorsal root. Okay, so this is the spinal cord, passing in through the gray matter there to synapse with the motor neuron. Okay, so one synapse right here. Then that information is converted directly into a motor or an efferent signal, which is sent out, exiting the central nervous system, and then causes the quad to contract. All right, and the reason that we have these primitive deep tendon reflexes is actually a protective mechanism, because if we were to overstretch the quad, then that reflex causes contraction of the quad to try to protect it from just passively tearing. Now with the polysynaptic reflex, for example, the withdrawal reflex, this is where you put your hand on a hot stove. That sensory receptor is going to send again that afferent information, right? It's going to be arriving at the central nervous system here through the dorsal root. But then this is where it's different, okay? So there's going to have to be some interpretation. Likely this is, you're going to be sensing either pain or you're going to be sensing extreme heat. sensory information is going to be sent in and is going to need to be processed by the brain. So it's not just a simple loop through. Although it looks like this interneuron is just connecting within the spinal cord, I want you to acknowledge that this information is likely going to have to pass on vertically directly up to the brain so that you can process either the pain or temperature signals or what have you. And then within that synapse within the brain, that's then you're going to have to make a decision, really quick one, which is going to cause you to likely withdraw in some way, withdraw your hand from that stimulus. The key is that the sensory is always in a polysynaptic going to synapse with an interneuron. Those interneurons then are going to connect that peripheral nervous system information to the central, and then the central nervous system is going to figure out what to do, and then will ultimately send out some sort of efferent signal here through the efferent neuron. And in most cases, that's going to be some sort of muscular contraction to get the hand off of the stimulus. That takes us to the end of the sort of first part of part one. So the rest of the lecture is going to be focusing in on the central nervous system, specifically taking a look at the brain. And we're going to be looking at the anatomy and the functions of different regions and how the tissue is actually set up. So a quick recap. And remember, looking at the cellular level, our nervous tissue is made up of neurons and glial cells, right? The neurons are the ones that are transmitting the electrical signals, okay? And we now know that a neuron's made up of dendrites, incoming information, the cell body, and then your axon, okay, which continues on to the terminal branches there. Okay, so cell body, dendrites, and axon. Now, all of those different parts of those neurons are then put together very differently when we're looking at either a peripheral nerve, which we'll see next week, or when we're looking at tissues within the central nervous system, specifically then the brain and the spinal cord. Within the brain and the spinal cord, you'll notice that all of the nervous tissue is organized into either white matter or gray matter. Okay, and you can see this visually. Here we have a coronal section of the brain, which is the same as a frontal section. And then here we're looking at a cross section of the spinal cord. Now, the white matter in both cases, here in the brain, you can see the white matter is located internally, whereas in the spinal cord, the white matter is located through the periphery. The white matter is always going to be composed of myelinated axons. Okay, that's what makes it white. Remember, myelin is a fatty tissue. So those myelinated axons are going to be transmitting information. In contrast, the gray matter then, which you see internally here in the spinal cord, and then you can see through the periphery of the brain, the gray matter is made up of everything else. All right, so let's go back. Okay, so what is everything else? Well, everything else, your cell bodies, your dendrites, okay, your terminal branches, and unmyelinated axons. Okay, so your myelinated you'll find in the white matter. Okay, your unmyelinated axons are going to be located within those regions of gray matter. In the peripheral nervous system, remember these axons are bundled together to form peripheral nerves, right? Within the central nervous system, however, these myelinated axons are organized into structures known as tracts, okay? And these tracts, I want you to picture that there are different regions where specific groupings of information are going to be sent either sensory up to the brain or motor from the brain down the spinal cord, okay? So these tracts are essentially columns of organized myelinated axons through which the information is going to be sent. So you have specific tracts for sensory information, and you have places where there are tracts for other types of motor information. Now, the brain is made up of about 100 billion neurons and 10 trillion glial cells. Remember, although the glial cells aren't transmitting any nerve impulses, they are so key to be able to myelinate, provide that blood-brain barrier, produce cerebrospinal fluid. The epidermal cells, it's another type of glial cell that produces cerebrospinal fluid, provide immune support, and so on. Okay, so both are essential to the overall function of our nervous tissue. Now, we also have different protective means protecting both our brain and our spinal cord. And the meninges, I'm sure you've heard of meningitis, right, which is inflammation of these meninges. Meninges are protective coverings that surround the central nervous system. Okay, so we have cranial meninges, which we'll look at today, and those cranial meninges surround the brain. And then we also have spinal meninges. And the spinal meninges, really, they're no different. We have 3 layers of meninges, so running deep to superficial, and remember that is important. So deepest would be the layer that actually covers the brain itself. It makes direct contact with the brain, and that is the pia mater. If you notice here, deep to superficial, the acronym is PAD, P-A-D. That's essentially what you're doing, right? You're wanting to pad to protect the brain. So that deepest layer is the pia mater. Then superficial to that, see this purple layer here? That is the arachnoid mater. Now, the arachnoid mater gets its name from the fact that it looks almost like a spider web. Then outside of the arachnoid mater, we have the dura mater, and the dura mater is the most superficial and it's the most durable. One thing I want you to note as well is there is no space outside of the dura mater. The dura mater itself in the cranial region attaches directly to the bones. Okay, and we'll see how this is different. When we look at the spinal meninges, we have the epidural space, right, which is where you receive an epidural. So we'll look at that as well when we talk about the spinal cord. All right, so deepest to superficial, we have the pia mater, arachnoid mater, and then dura mater. Now, between these layers, we have 2 spaces. So beneath the dura, we have the subdural space, and this is where you might have heard of a subdural hematoma before. And then below the arachnoid mater, so between arachnoid and pia mater here, this space is the subarachnoid space. And that subarachnoid space is essential for cerebral spinal fluid to circulate around the entire central nervous system, both the brain and the spinal cord. So the functions of these meninges: cover and protect the blood vessels that run through those subarachnoid spaces, the cerebral spinal fluid that's allowed to flow within the subarachnoid space, and also forming the different compartments. And we're going to take a look at ventricles a little bit later. If inflammation occurs, either due to bacterial infection or viral, it can end up with inflammation of the meninges, which is known as meningitis. A way to test for this would be through obtaining a lumbar puncture. And so here you're wanting to retrieve the cerebral spinal fluid to detect whether there's a bacterial infection or viral, and then that can determine the course of treatment. So with lumbar puncture, there's actually a video that I've included on our YouTube channel, and you can take a look there at the process that they would go through that. It kind of looks similar to how they would set up for an epidural. With an epidural, you're in the epidural space, so you're outside of all of the meninges. Here with cerebral spinal fluid being obtained through a lumbar puncture, you're needing to get through into that subarachnoid space to be able to get that CSF. Okay, so now moving deeper, we're going to take a look at the regions of the brain. Starting here with the diencephalon, which is this purplish region here located at the top of the brain stem. And then off the posterior aspect of the brain stem, we have the cerebellum. And then making up the bulk of, when you think of the brain, what you typically think of is the cerebrum. So first off, the diencephalon is made up of 3 main structures. You have the thalamus, the hypothalamus, and the pineal gland. The thalamus acts as a relay station. Back in the day, we used to actually go out to coffee shops and sit and talk to one another, right? And you would be a one-on-one conversation with lots of conversations around you, right? Your ability to be able to focus your attention on the conversation that you're having, part of that ability to just filter out everything else is regulated by the thalamus. Okay, so that's one way I want you to think about the thalamus. Now, the hypothalamus, we've talked about before. Hypothalamus acts as your main command center. Okay, we're going to look in the endocrine system. You're going to see how the hypothalamus is regulating all of your endocrine system, essentially. Okay, so all of the hormone production that occurs to regulate different processes throughout the body, that's all regulated by the hypothalamus. Now, also, we've talked about the hypothalamus's role acting as your internal thermostat. Remember, with negative feedback mechanisms, when the body temperature is going up, it's nice and hot outside, you start sweating, right? That whole negative feedback mechanism is all regulated by the hypothalamus acting as your main internal thermostat. All right, the hypothalamus is also responsible for a lot of your primitive drives. So when you're feeling thirsty, when you're hungry, sex drive as well, all of that is driven by the hypothalamus. And so then lastly, the last structure of the diencephalon here off the posterior aspect of it is the pineal gland. And the pineal gland secretes melatonin. When the light starts getting dimmer, there's a reflex that occurs. So we receive less visual light, and that decrease in light actually signals the pineal gland to start to secrete melatonin. And then melatonin helps to promote sleep. Even with our, like with daylight savings time and changing up those rhythms, it can really interfere with our body's ability to naturally regulate those rhythms. Now, so we just looked at the diencephalon. We're going to move down now looking at the brainstem. The brainstem is made up of three main regions: the midbrain, most superior, the pons, and the medulla oblongata. And we're not going to go into too many specific details here, but it is essential that we connect the dots with some things that we talked about when we looked at the cardiovascular and the respiratory systems. So remember, both of those systems are under automatic control. Well, a lot of that automatic control is regulated here in both the pons and the medulla. We have the cardiovascular centers, which help to regulate heart rate. We have the respiratory center, which helps to regulate breathing. And then lastly, the vasomotor centers, which help to regulate blood pressure. Now, posterior to the pons, we have the cerebellum. And the cerebellum is one of the portions of the brain that is most susceptible to intoxication. Okay? So you're drinking too much alcohol, the first place that's going to be impacted is the cerebellum. And as a result, they test that. If they're suspecting, say like a police officer pulls someone over, they're suspecting that they're intoxicated, they're going to be testing those fine motor movements that are coordinated by the cerebellum. So things like, touching finger to nose, being able to walk tandem gait, so heel to toe in a straight line. All of those movements are all coordinated by the cerebellum. Now out to the cerebrum. The cerebrum has an outer region and an inner region. So we talked about before the gray matter and the white matter. So the outer region, which is known as the cerebral cortex, is made up of gray matter. Okay, so the gray matter is cell bodies, unmyelinated axons, dendrites, okay, all of that. And the inner region of the cerebrum is made up of primarily white matter. So this is all of your myelinated axons. Now there are some exceptions where you have these islands of gray matter that you can see labeled here. Those are the basal nuclei. Deeper within the cerebrum, we have structures known as ventricles. And these ventricles are essential for producing cerebral spinal fluid. There are specific glial cells within the ventricles where cerebral spinal fluid is produced. Cerebral spinal fluid then is circulated from the lateral ventricles into the third ventricle, which then passes down to the fourth, and finally into the central canal of the spinal cord. Now, what we talked about previously was that all of those meningeal layers that are surrounding the outside of the central nervous system, between the pia mater and the arachnoid mater, there's that subarachnoid space. So once the cerebral spinal fluid has been produced, circulates through the interior of the brain and the spinal cord, it then passes into the subarachnoid space where it then circulates throughout the external aspects of the brain and spinal cord. Okay, so the ventricles not only produce cerebral spinal fluid, but are also responsible for helping to circulate that cerebral spinal fluid throughout the interior, which then connects directly with the exterior through the subarachnoid space. Now, some other external landmarks of the cerebrum, we have ridges, which are the elevated points, and then grooves, which are the regions sort of between those ridges. Okay, and they have specific names. So an elevated ridge is known as a gyrus, and those grooves are known as a sulcus. Okay, and each one of those identifies externally different parts of the brain that are involved in different processes. Now, right here through the mid-sagittal plane, running from the nose directly to the posterior occiput, you have the longitudinal fissure, and the longitudinal fissure divides the brain into left and right hemispheres. Okay, so when you talk about the left or the right side of the brain, it's the longitudinal fissure that is actually dividing those two structures. Looking within, if we were to take a coronal section, a frontal section here, this is what we'd be looking at. The white matter structure that connects the left from the right hemisphere is known as the corpus callosum. Remember, this is all myelinated axons, so responsible for simply connecting left hemisphere to right hemisphere and transmitting that information. As I mentioned before, those basal nuclei, which are those islands of gray matter, they're essential for automatic movements as well as regulating posture. we have the outer layer here, which is the cerebral cortex, remember, and this is made up predominantly of gray matter. Now, if we were to look from the external aspects here of the cerebral cortex, we have different lobes. Okay, I've listed 4, which I expect you to have the functions for, and I will introduce the insula, which is one other as well. Okay, so we have here, the great thing about this too is that these different lobes, they all coordinate with the different skull bones that these lobes are adjacent to. So you've learned that already. So here we have the frontal lobe. Okay, behind the central sulcus, we have the parietal lobe. Okay, here at the side of the brain, you have the temporal lobe. And then at the posterior aspect of the brain, the occipital lobe. The frontal lobe is responsible for motor coordination. Within the parietal lobe is your somatic sensory. Okay, so your sensory information is all processed here in the parietal lobe. In the temporal lobe, responsible for hearing. And then the occipital lobe, which is responsible for processing sense of vision. Now, coming back here, you see there's a couple of other regions here. It's region Wernicke's area and Broca's area. Both of these are regions of the brain that are specifically related to speech. But you can see they're located in different lobes. Okay, so this is just a good application, a good practice for you. So the temporal lobe, remember, is responsible for auditory sensation, right? So hearing. So Wernicke's area, if somebody was to have a stroke and it impacted this region of the brain, it would impact their ability to be able to understand language that somebody was speaking to them. Here with Broca's area, remember this is in the frontal lobe. And what's the frontal lobe responsible for? Frontal lobe is responsible for motor, right? So Broca's area, if somebody was to have a stroke that impacted this region of the brain, they would have difficulty in expressing what they wanted to say because the motor ability of them to be able to produce speech would be impacted. One final region here I just want to mention is the insula. So if you were to take this cerebral sulcus here, the lateral cerebral sulcus, and you were to pry it open from either side, and you were to look deep into the brain, you would see the insula. Okay, so it's a deeper structure. The insula is where we perceive pain. Big takeaways here, know those 4 lobes, frontal, parietal, temporal, and occipital, and know them in function. So frontal is motor, parietal sensory, occipital visual, and temporal auditory. And that takes us through to the end of this module. If you have any questions that come up in the meantime, please send me an email. Otherwise, take care.